What are some of the objectives for it? What I see is some key scientific and practical considerations. I'll give an example of a biomarker in which we as a field have made significant progress, an EEG biomarker, and then I'll talk about some challenges that I see in really getting to a stage where they could be useful in our clinical work and in clinical research. And then I'll talk about some strategies to do this. One being more rigorous research, and I'll talk about the work of the ABCCT, and then I'll talk about some more novel things that we're doing in the lab to try to think about how we could use biomarkers, how we could leverage them for care. So I want to start by making a really broad point. I said I'm a scientist and I said I'm a clinician. When I'm a scientist in my lab, I'm going to show you some of the different kinds of tools that we have to understand autism. EEG, I'll talk a lot about this today, electroencephalogram. This is an example of a positron emission tomography scanner that lets us measure actual molecules and synapses in the brain. This is functional near infrared spectroscopy that lets us look at metabolism and blood flow in the brain to see which regions are active. And this is eye tracking, which lets us look at a moment-to-moment basis what visual information a person's nervous system is taking in. So what an amazing array of powerful tools to make scientific inferences. But when I put on my other hat and I go to the clinic and try to make clinical inferences, I have a really different set of tools. And frankly, I have one tool. I have my clinical eyes and I have my clinical judgment. And that's the same tool that we've been using since the dawn of autism's time, when Leo Kanner first wrote about autism in 1943. And we've come a long way with our clinical eyes, but I think there's limitations on how much we can do, because there are probably elements of autism as a medical condition that are relevant to treating, to diagnosing, to predicting outcomes that we are just never going to be able to perceive with our eyes alone. So what would we want? Well, what I think we would want is biomarkers. What biomarkers are and this is biomarkers is something that's bigger than autism, and it applies to many conditions, from cancer to Alzheimer's disease to autism. And the Food and Drug Administration has brought together the National Institute of Scientists who really have expertise in biomarkers, and this is their definition: a defined characteristic that's measured as an indicator of normal biological processes, pathogenic processes, or responses to an exposure or intervention, including therapeutic intervention. So lots of words, but what do you take away from this? A biomarker is something that can be measured objectively that tells us something about an organism that's relevant to the comprehension or care of that organism. And so really what's critically different when we think about biomarkers from the clinical judgment that I described is that biomarkers are, one, objective, and two, theoretically, they're going to be more sensitive, right? And this is an example of things that we can't do. As a clinician, I'm going to see a child on Monday, and I'm going to have 45 minutes to look at that child and to interact with that child and to make inferences about their social behavior and communication. And they may not do certain things during that time, or maybe certain things maybe they'll be shy because they're in my strange doctor's office. And these are the things that, these are some of the limitations of clinical judgment. When we talk about biomarkers, there are many different kinds of biomarkers, and they can serve many different purposes within autism. Here are a few key examples. A biomarker might have prognostic value. Maybe measuring a biomarker in a person with autism today can tell us something about their course, what they're going to look like, the course of the autism, how it will unfold over coming years. There are biomarkers that are called either pharmacodynamic response, or another way of putting it would be like a target engagement biomarker. That if you thought a medication, or if you thought even a behavioral treatment was expected to affect a neural system, it might show you that neural system is being engaged, right? That would be useful. So the third kind of biomarker, and I think this is the biomarker that people think of most when they think of autism, is a diagnostic biomarker. Now, when I say this, I bet that almost everybody rightfully thinks this diagnostic biomarker tells me who has autism and who doesn't. And I think it's a way that we tend to think about biomarkers. And I feel like 90% of the time, most people in the field of autism hear biomarker. They think, oh, they're talking about a diagnostic test. And it doesn't need to be that way. And I'll make the case that it shouldn't be that way, because when we talk about autism, we're not talking about one biological entity. We are talking about hundreds, maybe thousands of biological entities that just kind of look similar phenotypically to the clinical eye, and that's how they ended up there. And maybe it's unrealistic to think that we are going to find some biomarker that is definitional of all those different etiologies. But we can also think about biomarkers that are diagnostic in a different way. We could think about them as biomarkers that are diagnostic of subgroups of autism. The idea that we have this extremely heterogeneous, diverse group of people with autism, and the biomarkers, maybe they're not going to say the way those people with autism are different from everybody else on Earth, but maybe they can help us understand the way people with autism are different from one another in meaningful ways. Maybe these people that are greener, we're going to benefit from a certain kind of treatment, and the oranger are going to benefit from a different kind of treatment. And a biomarker could help us do that. And that would be extremely useful. And I also like to highlight this as an example of where biomarkers could maybe help us where where our clinical eyes have failed us, and that we have tried for many years to come to subtypes of autism based on our clinical judgment, and as a field walked away from it in 2013. Right, we had Asperger's syndrome, we had PDD-NOS, we had autistic disorder, and the field made a determination those aren't working. And so it's an example of maybe we can be certain that there are subgroups of people with autism that would be good to know. We can also be certain that we haven't found them yet based on clinical judgment alone. And this is an example of where biomarkers could potentially be very useful. I can't see the participants, but there's someone who's unmuted, and I'm hearing some background noise. If you can make sure that everybody be muted, that'd be very helpful. Thank you. When I think about the scientific, what are some things that I think are scientifically critical about biomarkers in autism? So this is you might think of it as like kind of my list of priorities, and I'll make the point, it's a nuanced but extremely important point that I want you to take away from today, is that a biomarker doesn't have to do everything, right? Like I said, there's prognostic, there's target engagement, there's diagnostic biomarkers, and a biomarker doesn't need to perform the same way to do all those things, right? Like if you had a treatment biomarker, you would want it to move up and down as a person got better. But if it was a diagnostic biomarker, you wouldn't want it to move around every time a person got treatment, right? Otherwise, people would be hopping on and off the spectrum. And so these are the kinds of things that I think are relevant to understand. But I want to make the point that a biomarker doesn't need to do all these things to be very useful for one purpose, or the FDA term is context of use, for one context of use. So these are things that I think that a biomarker should be sensitive to diagnostic status. I don't mean to go back on what I said. To be sensitive to diagnostic status isn't to define diagnostic status. But I think if we're not, at least on average, seeing some differences in biomarker values between people with a diagnosis and people without, probably low likelihood that it's telling something meaningful about autism. Relatedly, maybe some biomarkers we don't want to be thinking about the level of diagnostic group, but maybe thinking about them in terms of particular symptom domains. That we might want biomarkers that track with speech, or biomarkers that track with face perception or sensory processing. And in those cases, we might want to see that we might be less interested in showing that the biomarkers associate with the diagnostic group, but they associate specifically with that symptom domain, that the biomarkers are tracking those functional processes. And relatedly, we would want to understand that they're not tracking things we think they're not measuring. And let me give you an example. If we had a biomarker that we thought was relevant to social, say social function, but little do we know that it's actually tracking with intellectual disability. If we just measure social function, it's going to look pretty good, because intellectual ability is going to cap social ability in some way. So we would see meaningful relationships if this biomarker looks different in people who are less social. But then what if we treated those kids so that their social skills got much, much, much better? Well, we thought the biomarker was tracking social skills, but it's actually tracking intellectual ability, which isn't being affected by this treatment. So we're going to see, even when the social skills are improving, the biomarker is not moving. And so what we want to know is, what the biomarkers are associated with, but also we want to know that these associations are specific. We want to understand how these biomarkers are relevant across development. This is a real challenge for the field of autism. This is not a challenge for people who are doing biomarkers for Alzheimer's disease, right? But here, when you say autism, you might mean a person who's 3 years old. You might mean a person who's 30 years old. You might mean a person who's 60 years old. And our brains look structurally and functionally very different at all those different age points. And so when we talk about biomarkers that are indices of brain function, we may be able to use the same biomarkers across development, but we may need different biomarkers for different developmental windows. And that's okay, but that's something that we need to be thoughtful and planful about. We need biomarkers that are robust to variation in behavior, depending on the kind of biomarker. Let me tell you what I mean. If we have a genetic biomarker, this doesn't matter. If a child is enjoying a blood draw, or if a child is extremely upset during a blood draw, the genetic information that is extracted from that blood is going to be the same. However, if I'm going to talk a lot about EEG research today, if I want to do an EEG biomarker looking at speech or something, if that child is screaming and crying during the EEG, well, that's the only signal I'm going to get, right? So the way a child is behaving, the things they're paying attention to during biomarker acquisition for functional brain-based biomarkers can totally obscure what you think you're measuring. And so we have to be thoughtful about that. We would want biomarkers. We'd want to be one to understand that biomarkers might be sensitive to change in clinical status, that if a person gets better or if a person gets worse, that we see the biomarker values change. I think this is another great example of a place where biomarkers could do something that we don't do well in autism at all right now. Right now, if you want to know if a person has gotten better in response to treatment, and actually this is talking to a group who probably thinks well about this, and that there's probably lots of BCBAs who actually measure behavior in ways that you could do it very meaningfully. But in much clinical research that's not kind of behavior analytic, the way you decide if treatment is working is you hand a parent a questionnaire that asks about these socially abstract behaviors over the course of 6 months. And if you maybe just did a 6-week treatment, what does it even mean to fill out a questionnaire that's asking about 6 months to measure treatment that happened in the course of 6 weeks within those 6 months? So this is another place where biomarkers can make a big difference. So these are the kinds of scientific things that I worry about. I worry about biomarkers, and I mean worry in a good way. I also worry about practical things because I don't just want these biomarkers to be useful in my lab. I want them to be useful in gigantic clinical trials, and someday I want them to be useful in clinical practice. So if we think about things in those terms, there's practical considerations that are relevant. One, it needs to be viable in this group. So we need things that can be done safely in children and that children can tolerate, right? That sounds obvious, but there's many studies done about biomarkers that might be produced with functional magnetic resonance imaging. And I think there's a lot of kids won't ever be able to tolerate those kinds of procedures. And so there might not end up, it might be a ceiling on how useful it could be. We also, relatedly, if we can pick on fMRI today, in terms of cost effectivity, if we're going to do biomarkers at scale, we want the collection of that information to be cheap. And so if we had a technology that costs $500 to run it on every person, it wouldn't make any sense to run it on everyone if we're expecting it to be informative for a very small subgroup of people that we run it on. And then lastly, we want biomarker technologies that are accessible, that if I had a great technology that could be extremely useful and was cost effective, but there's only one machine that exists in my basement, right? It's not going to be too useful. And as an example, like if we had a technology that could be used at NIH Autism Centers of Excellence, this would be how many people it could reach. If we had a technology that could be deployed at every hospital in the country, this would be how many people it could reach. And so this is obviously what we're more interested in pursuing, accessible biomarker technologies. I'm going to talk a lot today about EEG. EEG is the electroencephalogram. It's a way of measuring brain activity by recording the actual electricity that neurons produce when they fire. And when many neurons fire in unison, the signal together is actually a strong enough electrical charge. It's minuscule, but strong enough that you can pick it up from a person's scalp. And that's what the EEG is. And so the way we do it is we have a person wear one of these they're called EEG nets or geodesic sensor nets. It's an elastic mesh with small rubber pedestals embedded in it. Inside each pedestal is a sponge, and at the base of that sponge is an electrical sensor connected to a wire that then connects to an amplifier and a computer. And what we do is we take this whole elastic mesh and we dip it into a salt water solution so the sponges absorb that salt water and become conductive. And then when we stretch it over a person's head, the sponges make contact with the scalp and form a conductive bridge so we can record the electrical activity. It doesn't do it from the whole brain, from really the outer layers of cortex. When we do EEG recordings, generally we look at two things. One, what we would call resting EEG, which is just the idle of the brain. So when we're not really making a person do anything, their brain is of course doing many things. And what we see is there's oscillations at different frequencies, and the different strength of those signals and the different balance of signals across those frequency bands can be informative in understanding the way people with autism, their brains at rest, differ from other people. We can also do what are called event-related potentials, or ERPs. And what this means is when you're doing the EEG recording, you make specific and discrete perceptual events happen, like you flash faces on screen, or you blurp sounds into the environment. And then what happens is the brain responds to those things. And if you do it many, many, many times, like maybe 20 to 60 times, you can see what is being produced uniquely by that signal. And then that gives you an index of the specific cognitive process that's induced by what you just made happen. So those are the two ways we look at EEG. One of the things that's really nice about EEG is for people interested in understanding autism and for looking at it in children, it's a really, really user-friendly way of measuring brain activity. So we can do this in infants, we can do this in people who have genius level IQs, and we can do it in people with significant intellectual disability. It's non-invasive. The technologies that we have now don't even require you to abrade someone's scalp, and it's movement tolerant. If a person moves around during an EEG recording, the particular times when they're moving around we won't get meaningful information from, but the rest of the recording is still intact. And so compared to other technologies where if you move during a scan, you might not be able to co-register the functional information with the structural information, EEG is pretty useful and user-friendly technology. When we think about some of the practicalities I mentioned, EEG is also a really great technology. It's expensive to get an EEG system, but once you have an EEG system, it's really, really economical to acquire data. It costs salt water and latex gloves. So it's really cost effective. And EEG is also really accessible. So the particular EEG system that I use isn't everywhere. It's really used mostly by researchers. But if you think about, if we figured out something here, there's no reason it couldn't be measured using any EEG system. And right now, every hospital in the country has an EEG system, and it's already used at the population level for screening. We use it to screen for seizures. We use it to screen for hearing problems in newborns. And so if we should learn things about autism that are meaningful, EEG is a great technology to be able to deploy at large scale in a realistic way. And lastly, EEG is really, in autism, there's many, many ways you can try to understand autism, right? It affects so many different systems and so many different functional processes. The one thing you can, you've heard people say, if you've seen one person with autism, you've seen one person with autism. That's kind of true, but I actually think what's more striking to me is if you've seen one person with autism, you've seen a person, you don't know their IQ, you don't know their language level, but you know that they have difficulties with social communication. It's the only thing that's true, diagnostically speaking, of everyone with autism. And EEG is a technology that's been applied really productively in typical development to understand social communication. So when we come to understand autism, which is, as we've said, is a heterogeneous and diverse group, we've got a strong foundation of knowledge in what is the one unifying area of relevance for everybody with autism. And this is an example of a key thing we've come to understand about social communication using EEG. So this is an event-related potential, or an ERP. This is the electrical activity that you see when a person sees a face. And what you're looking at here is on this x-axis is milliseconds. So this is really, really fast. So these are milliseconds. So, this whole chart is 7/10 of a second. These on the y-axis is the strength of the signal, which is measured in microvolts, which is very small, so 1,000th of a volt, right? And so these are not like you're powering a battery or something like that. And what this is highlighted in purple is called an N170. And what that means is the N is for negative. It's a negative going electrical pattern of deflection. And 170 is its timing. It happens at around 170 milliseconds. And what we know from many studies in typically developing people from infancy through adults, that this is the brain recognizing a face as a face. And we think that's important for understanding autism because faces are such a key source of social information. When we look at the N170 in autism, and we did the first study to do this now in 17 years ago in 2004, we found, we didn't know whether people with autism would show an N170, and they did. We found that it was different in a really significant and important way, and that it was slower. So we saw that people with autism were inefficient in their brain's recognition of a face as a face. And we think this is important because when we see a face, when you recognize a face as a face, your brain's still got a lot of work to do. It's got to figure out who it is. It's got to figure out what it's saying, what emotions it's experiencing. And even at that very, very first stage, people with autism are already operating at an inefficient pace. And so we think that could contribute to some of the difficulties that they have with face perception. I'm going to tell you a lot about the studies that we have done to understand the N170 and what it means for autism. And as I do, I'm going to keep referencing back to that list of scientific considerations that I described. And so here I'll reference back. We see it's sensitive to diagnostic status. The N170 is slower, more inefficient in people with autism relative to typically developing individuals. As a part of the study, we also wanted to understand how this brain activity that we're measuring in the lab might relate to things that are more obviously relevant to real life. And ability to recognize faces is something that's very relevant to real life. So we administered a test, a neuropsychological test of face recognition called the Wechsler Memory Scale, or for children, the Children Memory Scale. And we found that people with autism made a lot more mistakes, twice as many mistakes about as they're, even being IQ matched as the typically developing adolescents and adults, and that how good they were at face recognition was related to the speed of their N170. So again, to reference our list, we're seeing that the N170 is also associated with symptomatology and function in a behavioral domain relevant to what we think we're measuring with the biomarker. The next thing that we wondered was, okay, so we're seeing the N170 is slower, but is this telling us something about social communication? Or what if the brains of people with autism respond slower to everything they see? Then it might still be useful to know, but it might not be telling us what we think we're understanding about social information. And so that's tricky to measure, right? What we want to show is that N170 works fine for something else. And so we got an idea from a boy that I was seeing in the clinic. So let me show you a really quick clip of, and I apologize, it's less interesting to look at, but because it's being video recorded, I obscured his face.
These W's look like they're on top of each other.
They sure do. You a picture.
A picture?
Yeah.
WPS?
Oh, that's just remember that.
Hey, That boy's not underwater.
He's got a mask and a snorkel.
And it looks just like a J.
You're right, it does look like a J.
Okay, so you see this video. What's this guy interested in? If we were all in a room together, I could say, what's this guy interested in? You would all shout out all together, letters of the alphabet. Right. And it turns out a few things. One is that even though language is a primary area of difficulty for many people with autism, letter decoding and reading is actually a strength, that people with autism tend to read as well as you would expect based on their IQ, even though they have difficulty with the reading comprehension. I mean, the physics, the orthography and things like that. And so it also turns out that when you learn to read an alphabet, it doesn't matter what alphabet. The experiment, the first experiment to find this looked at Chinese and English. You get an N170 to the letters of that alphabet, reflecting that your brain is specializing to read the letters of that alphabet. So we got an idea. We could do a test. We could say, all right, we've seen that people with autism show a different N170 response to faces, and we think this is meaningful about social, but let's compare it. Let's compare their brain response to letters, and then to understand whether they were showing specialization in the N170, we compared it to fake letters, pseudo letters. And so basically we're saying, if we see differences in both places, well, then there's nothing special about the social information. Just people with autism are slower to process visual information, and we should understand it as that. But if we see differences here, but not here, well, then that would support our inference that this N170 is telling us something that's really specifically meaningful about social information. Now, we also thought, when we did the study, we thought, hey, you know what? We've seen these N170 differences in adolescents and adults. Let's use younger kids and see if the N170 is also true in this developmental cohort. So it gave us a chance to look at the developmental range to which these N170 differences might apply. And when we looked at the social stuff, we saw the exact same things that we had seen in the older people in these kids. We saw that the people had more problems with face recognition, and we saw their N170 was slower among people with autism. And so this delay seems to be there, across a big chunk of the lifespan, children, adolescents, and adults. When we looked at the non-social things to see whether these difficulties are specific to the social domain, well, in terms of behavior, people with autism did just fine. Their word reading and word decoding scores were in the average range and were the same as the typically developing children in the study. And then when we looked at their brain responses, their N170 to letters, they also looked just fine too. We saw that what this chart shows is these purple bars are letters, the green bars are the fake letters or pseudo letters, and you see that everybody, whether you have autism or not, is showing this big difference of specialization for the letters compared to the pseudo letters. And we don't see any differences in latency, which aren't pictured here, but they're not slower for letters. And so what this tells us is that this N170 slowness that we see isn't just saying, okay, autism, slow processing. It's saying autism, different, slower processing for faces. So this dissociation is telling us that the N170 is measuring what we think it's measuring. Next thing we wanted to understand was we wanted to kind of rule out that what we were seeing, that we thought we were seeing in the brain, might actually just tell us something about the way people with autism look at faces. Because when you look at a face, if you look to the eyes of the face, your N170 is faster. And we know from eye tracking studies that people with autism tend to look less to the eyes when you show them an on-screen face. And so maybe that's all that's happening. Maybe we're not actually seeing a difference in the N170 response that's meaningful. Maybe when we do the experiment, people with autism are looking to the mouths, and so we're seeing a slower N170. So we did an experiment to look at that. And we used crosshairs on screen preceding faces to control, so that we were making a person look to the eyes, making a person look to the nose, making a person look to the mouth. And what we wanted to understand was, if the person looks to the eyes, do the differences that we've seen in the people with autism go away? Or, what we what we hypothesized is that no, this is telling something about the brain. It's not just about what visual information is getting to the brain. It's something about the way this information is being used in the brain. And so even if you make a person with autism look to the eyes, they're still going to be slower. And that's what we saw. So let me walk you through what you're looking at here. So the yellow dots are individual N170 latencies for people with autism, and the blue dots are for typically developing people. These two columns are when they're looking at the eyes, the nose, and the mouth. And what I want you to see is that when we force a person to look to the eyes, rather than that speeding up the people with autism's N170 and making them look more similar to the typically developing people, that's where the differences are most pronounced. Is that we see what we expected, that people with the typical development show a faster N170 to the eyes, but people with autism do not. Moreover, this is where the difference tends to exist, right? The people with autism are rather than looking to the eyes, getting rid of the difference in the N170, that's where it's most pronounced. And so we see that this N170 delay exists in autism even when we're controlling for attention to the eyes. And so we're saying it's not an artifact. The N170 difference we see is robust to variations in behavior when we're acquiring data from people with autism. And then the last thing that we looked at about the N170, and these are really preliminary data. We had an opportunity. Our colleague Pam Ventola at the Child Study Center is doing research looking at a behavioral treatment called pivotal response treatment. It's the idea of this treatment it's a naturalistic behavioral intervention in which you teach children core social skills or pivotal skills, and you practice them in the context of play, so they find these things to be more reinforced. And the idea being is that that's going to motivate them to use them in real life and kind of snowball, that they get these skills and they get more enjoyment out of social interaction, and their social skills grow. And it seems to work. There's a number of studies that have shown that pivotal response treatment is very effective. And so Pam was doing this and having kids come in for a 14-week course of treatment. And we thought, what if we can take a look at the N170 when the kids come in, and then after they've received their 14-week course of treatment, to see if as kids get better, if their N170 gets faster, if it moves in the direction that we would think is more typical. And that's what we saw. And I say these data are preliminary because the sample is so small, because we weren't able to do elegant things like have a good like a placebo. You can't really do a placebo control for this, but like have a treatment as usual condition or a waitlist condition. So these are not strong data, but they are consistent with what we would think. So you can see here individual data from 7 kids that participate in this study, and you can see that this is the before N170, this is the after treatment N170, and you can see that every child except for child number 3 is getting faster. Their N170 is getting faster as they get better in treatment. And I'm not going to show you the behavioral data, but all the children did get better during treatment. And so that's exciting to us. I mean, obviously more needs to be done to understand this, to understand things like why child 3 didn't get faster, and to what degree the changes in the N170 parallel or associate with changes in behavior. But it's suggesting to us that the N170 latency might also be useful in terms of indexing changes in clinical status. Let's revisit my list of scientific priorities for biomarkers and think about the N170, what we've learned. So we've seen the N170 is sensitive to diagnostic status. It's slower in people with autism. We've seen this associates with symptoms like face recognition, difficulty with face recognition. We've seen that it's functionally specific. It's not just telling us that a person with autism is slower at processing everything. And we've seen that these differences are applicable across development. We've seen them in children, adolescents, and adults. We've seen that it's at least for gaze behavior, these differences are robust. It's looking to certain parts of the face don't cause or eliminate this difference. And we've seen, in a preliminary way, that it may be sensitive to change in clinical status. So these are all great things that we would want to see for many biomarker purposes in a potential biomarker. We also could think about our practical considerations. Remember, because we're using EEG, the N170 is something that's viable in populations with special needs. It's cost effective and it's accessible, right? So The N170 has all these things going for it. Why aren't we in a place where we can use this for some of these biomarker purposes that I described? Why can't we use this as the outcome measure in a clinical trial? Why can't we use this in a clinic to show that a kid is doing better in treatment? There's a few things that really need to be ironed out before we can do that. I also want to make the case, I've told the story about the N170 because it's what I'm most involved with, but this is not a story that's unique to the N170. There are many biomarkers that show promising evidence in many of the same domains that I've described for the N170. And they are also, they also have the same problem as the N170. They're not there yet. They're not ready for being used. And part of the reason why is because the differences that I've described to you aren't universal truths. In science, in the scientific methods, we would call this replicability or reproducibility. We don't see the same results all the time. We don't know why. So at the bottom of the slide is every study that I'm aware of that has followed up on our first finding of that N170 in 2004. And many of them found the same thing that we did. In fact, what's cool is this one, Kang, is actually a meta-analysis that took all of these studies and put them into the meta-analysis sausage grinder and said, whoa, on average, across all these studies, this is true, that the N170 is slow. But some of these studies didn't find that, and we don't know why. And that's really important to understand. One reason could be is that that's just the truth, right? People with autism are all different. Why would we expect that a biomarker value that isn't definitional of autism wouldn't be different across samples? Maybe some of these samples were really hit in their face processing system. Maybe some were really intact in their face processing system. And maybe that's maybe the N170 is telling us something about this specific system and not about autism as a whole. And that's fine. That could be really, really useful. But we want to know that. That's a good problem because that we can tease out. We can do studies and look at those relationships and understand it. There are harder things about sussing out differences in this literature. One, many of these studies are small, and so they could have yielded false positives or false negatives. And there's really nothing that we can do now going back and looking at these studies except be more responsible in future research. And we also don't know how methods could affect results. So of these studies, there's many different EEG systems used, many different kinds of face pictures, right? Color, black and white, emotional, neutral. And we don't know how those things might affect an N170. So there's lots of kind of methodological things that could contribute to these differences in reproducibility. And we want to understand what's what before we could use them practically. There's other things too, like for a biomarker to be used in the context of a clinical trial, if you're going to, by definition, if you're going to be using something at multiple time points, we want to know that it's reliable within a person, that a person's value today tells me something about their person's value tomorrow. And we also want to know whether the act of repeatedly measuring it changes it. Like what if doing EEGs makes your N170 faster? Then that might explain our preliminary results about PRT, right? Just because there were 2 EEGs, their N170 got faster. We don't know those things for most potential biomarkers in autism. Another challenge is that we don't have a normative reference. By that, I mean, like every time we do a study of a biomarker in autism, we have to have a control group, and all we know is how our autism group compares to that control group. Contrast that with head circumference. Whereas if you're looking at head circumference in autism, you can go to the CDC website and download growth charts that tell you, and you can know for any given person with autism exactly what percentile rank their head size is. Like we don't have things like that. And so most of our understanding of biomarkers in autism is relative. What do? We do better studies. We need more rigorous studies, and I'll tell you how I would operationalize more rigorous. And really, this is on the FDA's radar, right? The FDA wants to have qualified biomarkers that could be used in clinical trials, that could be used by industry for these purposes. So what should we be doing? Well, we could take biomarkers that are pretty well studied and study them in more powerful ways by using really, really well characterized cohorts so that we have the individual information to understand the relationship of the biomarkers to the subtle aspects of the clinical phenotype. We would want to have big samples, including big samples of typically developing children, so that we have the statistical power to look at these relationships. in a longitudinal design so that we can look at reliability and change over time. And we'd want to do it methodologically tight so that we're not worrying about whether different EEG systems or different stimuli are confounding our results. And we'd want to do it with practical assays so that if what we learn is useful, it could be rolled out potentially. And these are all the considerations that led to the genesis of the ABCCT, the Autism Biomarkers Consortium for Clinical Trials. So we are now in our sixth year. I'll tell you what's happening now, but first I'm going to tell you what has already happened. So this is a multi-site study based at Yale, spanning 5 autism centers around the country. Our first phase, we conducted a really large biomarker study, 280 children with autism and 119 typically developing children, school-aged, IQ spanning intellectual disability to well above average. We used practical assays like EEG and eye tracking and a longitudinal design, seeing kids at a baseline at 6 weeks and 24 weeks. And we also drew blood. Now, the ABCCT wasn't actually funded to do any genetic analyses, but we have that information, so we'll eventually be able to look at how our biomarkers relate to genetic information. Other unique things about the ABCCT, it's a funding mechanism called the U19, which brings together a really, the leadership structure is I'm the principal investigator, but I run it with a steering committee. And that steering committee has scientists from NIH. We work closely with scientists from pharmaceutical companies. We work closely with scientists from the FDA. We work closely with scientists from other consortia that are designed to develop biomarkers. And in this way, we have a tremendous amount of expertise being brought to bear on the study, like the idea that we can do it as well as we could possibly do it with all this expertise. We did this with an unprecedented level of rigor in terms of the way we run the study. We run it as if it were a clinical trial, a set of procedures called good clinical practice. Methodologically, bulletproof, or as bulletproof as one could be. Every site used the same EEG system. You know, we worried about things like our data acquisition team built custom light meters so that we could be sure that the ambient light in every EEG lab was the same. So we worried about everything to try to make sure it was getting as much signal as we can, as little noise as we can. And then lastly, statistically, we were really rigorous about pre-specifying things. So no messing around saying, hey, let's take a look at that, let's explore that. Before we did any analysis, we said, this is what we predict. We're going to pick one biomarker that's going to be our primary. We're going to make a directional hypothesis about that. And the last thing we did was we coordinated with a European network called EU AIMS so that we would actually both be doing some of the same biomarker assays so we could compare our results in 2 independent samples. The next few slides, I'm not going to really go through in detail. They're there. I'm happy to share my PDF. And if you want to know these things, we did, suffice it to say that these are we did the gamut of clinical measures currently used. We did 4 EEG and 5 eye tracking measures. And what I'll talk about is you would assume one of the EEG measures was the N170 that was actually the one we designated as primary. And that asterisk means it was also one that was administered in the European consortium. What did we learn from studying the N170 in this big study? A few things. First is the findings hold up. So we see that the N170 is slower in people with autism than typically developing people. 2, we learned that we can require this information pretty reliably. We got valid signal from 97% of the typically developing kids and 3/4 of the kids with autism. 3, this information is pretty stable. So this is what's called an intra-class correlation coefficient. It's basically how well a person's values hang together with their own values over time. And we see, what we would call adequate reliability, 0.66 to 0.75 at 6 weeks and at 24 weeks. We also see that the kinds of things we've seen before, the N170 is associated with face memory. And we saw that we saw some predictive value, that your N170 today tells me something about what your face recognition skill is going to be like in 24 weeks. So this is important. This is a high profile replication of a neuroscientific finding in autism, which doesn't happen all the time. What we did with this was approach the FDA. They have a biomarker qualification program, as I mentioned. The first step is to submit a letter of intent. We did so, and the N170, and I haven't gone into detail today, but an eye tracker biomarker called the ocular motor index of gaze to human faces, basically how often people look at faces on screen. Both of these were accepted into the FDA's biomarker qualification program, which is a milestone in that these are the first two biomarkers for autism and the first two biomarkers for any psychiatric diagnosis to be accepted into the FDA's program. It doesn't mean we don't have a long way to go, but it's still exciting to have made it to this point. What the way we've described their potential use to the FDA is as a diagnostic biomarker that is diagnostic of a subgroup, right? So this is the distribution. These are autism. These are typically developing kids. This is their N170 latency. And you can see it doesn't tell you who has autism and who doesn't, because if you have a value of 200, you're almost equal likelihood of having autism or having typical development. However, you can see that when you get to the slowest N170s, that's really an almost exclusively autistic group of people. And the idea is that maybe this tail represents some more homogeneous subgroup of people with autism, and that if we selectively use those people in clinical trials, we might have more statistical power to find results because it's less biologically heterogeneous. What happens next is we prepare what's called the biomarker qualification plan, which is our roadmap for data analysis and data collection to get us to the final stage, which would be a biomarker qualification package. And this is an interactive process with the FDA. We've actually had two of these U grants to fund us to have discussion with the FDA, to think about things that are really hard, that nobody, not us, not the FDA, really knows exactly how to do yet for psychiatric conditions. Where do you draw this line for a continuous distribution? How do you prove that this purple group is biologically different from the people outside of the group? How do we ensure that the things that we've learned from this study are generalizable to other studies using other systems? And this is all work in progress. We're really excited that in July of last year, we were funded to do a second phase of the ABCCT. The second phase is going to consist of 3 studies. The first is a confirmation study, which is basically going to be a replication of that first study that I've already described to you. The only difference is we're going to take out one biomarker that didn't work very well, and we're going to change our balance of our sample. So it's like half autism and half TD, instead of being 2/3 autism, 1/3 TD. Second study, and it's funny, I'm describing them out of sequence, and the confirmation study will start next year. The follow-up study is starting right now, and that is to bring back that original cohort of children that were in the first study to look at long-term biomarker stability, to see whether if these kids have gotten better or worse in the last 2.5 to 4 years, if their biomarkers have proportionally changed, and to see whether the biomarkers measured now, several years ago, are telling us something about how these kids are functioning today. So that's the follow-up study. It's underway, and then the last study will be a feasibility study to see whether this biomarker battery is viable in younger children, in 3 to 5 year old children. And I'm going to show our contact information in my last slide, but I beseech your help in that when you see this confirmation study, to bring in 400 kids in a short amount of time for 3 visits is hard. It's hard for some of our sites that are in Boston or Los Angeles or Seattle. But it's really hard when you're in a metropolitan area the size of New Haven. And the only reason that we were successful the first time is because of the hustle of our team, but also because of the collaboration of the community, teachers, autism support groups locally, just getting the word out, and families being willing to come in and work with us, because it is the number of kids and the time frame that we want to work with starting next year is really, really ambitious. And so I hope that you all will help us carry out this challenging but really important study, and I'll tell you more about how to help us in a few slides. I want to end, so the ABCCT is interesting, at least interesting to me, in that it's interesting in its scope and its scale and its rigor, but really what we're doing is shoring up understanding of things that are already pretty well understood. I want to end by talking about something that's the opposite, is kind of going out on a limb and doing something that's innovative, high risk, high yield, you might say. And so could we use these biomarkers to help us actually administer a therapeutic intervention? We know that right now we treat autism behaviorally. Most behavioral treatments target social function, and we know from a handful of studies that have been done um, that there are certain brain systems, like the superior temporal sulcus is an important brain region for social processing, that seems to change when behavioral treatments work. Well, what if we could shortcut the behavioral treatment and just turn on the STS directly? So there's a technology called transcranial magnetic stimulation. And what it is this paddle you see emits powerful powerful magnetic pulses, and they cause neurons to fire. And if you do this with a certain level of rhythmicity, you keep that portion of cortex kind of jazzed for a period of time. And there have been a couple of studies that have suggested that this actually makes social behavior better in autism and reduces some of the repetitive behaviors. This is really still a nascent area of research. The treatments were long. Our idea is, what if, instead of doing it for 8 weeks, what if we use what we understand about biomarkers to tell us in a much, much shorter time of frame whether it looks like this is going to work for a given person? Like, if we stimulate STS, could we see in some people their N170 is getting faster? Could we see whether they're looking more to the eyes of the face, which are both, the N170 and visual attention, we know to be related to function of the STS. And so this is a line of research that we're literally just starting. We're hoping that we're actually going to receive funding from the Department of Defense to start a study this summer. But in the preliminary work that we've done, which is really just looking at typically developing adults who don't have social problems, it does what we would think it would do. We see that after stimulation, their N170 gets faster. We see that after stimulation, they look more to the eyes of the face. And so it seems to be moving the needle with these biomarkers in the direction that we would hope. And stay tuned, because we're really excited about the potential utility of this to actually be a way to help people with autism, in the short term. I'm going to stop there. I want to thank, most importantly, the people with autism, kids, the adults, their families that devote a significant amount of time and hassle to traipsing into New Haven, to letting us put these elastic hats on their heads, helping us understand more about autism. I want to thank clinicians in the Developmental Abilities Clinic, because that's really where our work begins. That's how we connect with people with autism, and is the most important service that we provide to the community. I want to acknowledge the really broad group of people involved in the Autism Biomarkers Consortium for Clinical Trials. The leadership is shown here, but there's many, 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 many more people involved, and it's an outstanding and very hardworking team. And I want to thank the people in my lab who really carry this work forward day to day. And you can see our contact information there if you want to learn more about what we do. And as I mentioned, we need your help. Tell people what we're doing. Tell people that that they can help us understand more about autism. We've got many exciting studies going. I'm happy to say that we in the lab and in neuroscience research have a long history of excluding people with autism who also have significant intellectual disability. And we have studies going now that are specifically for people with autism and intellectual disability, or people who have intellectual disability and not autism. So really nowadays we can work with everybody. We will pay you for your time, and you'll be evaluated by our really outstanding clinicians, and you'll get a report which we hope will be useful for you in school. We're all vaccinated. We've rebuilt all of our labs to be extremely safe in the era of COVID, and so please spread the word. We're doing research, and we want people to do it with. And I'll stop there, and I'm happy to answer any questions or field any queries if there are any.
Fantastic talk. Let's see. I hope people can hear me now. I think I fixed the damned internet. Jamie, that was great. Do you have time for a few questions?
I do. I'm yours.
Jamie, I did have one question come directly to me asking where people could get the treatment, and they wrote STS treatment, but I think it was the ITBS treatment. And would it work with intellectual disabilities?
So for now, we're in the very first phase. I wouldn't even call it treatment yet. I would call it more research to see whether it could be therapeutic. And the first study that we're doing is only in adults, and it is only in adults with average IQ or above. That is a requirement of the IRB at this stage because it's a new technology. But if you're interested in getting within those constraints, please contact Erin and she can tell you more about eligibility criteria for the study. And I can see a question in the chat. Do we have any autistic adults on the steering committee for the ABCCT? We do. A man, John L. Robeson, who may be known to many of you, is on our advisory board, as is Allison Singer, who's the head of the Autism Science Foundation and is also the mother and sister of people with autism. And then John, who many of you may be aware, wrote a book actually about his experience with TMS, also advises us on our use of our TMS study. So we try to operate with the input of people with autism in our studies. Another question that came into the chat is, in your studies with autistic, in your studies with adults, have you been able to make any difference between adults diagnosis, children versus late diagnosis? We have not. We've never. We have not even looked at that. We have not looked at that as a dependent variable in any of our analyses. It's an interesting idea to look at early versus late diagnosis off the cuff, speaking not as a scientist but as a clinician. I would say obviously, the people with late diagnosis are people who are probably going to, passed or camouflaged for a good function of their life, and so who may show milder effects in some degrees, although I use the term mild with reservation because some of the other things that happen, like anxiety and depression, can be extremely significant and severe, irrespective of, for example, your fluent use of speech. Another question is, I work with autistic adults and I'm curious how you present your work to individuals who may be skeptical about treatment for autism, especially when autism is part of their identity. Well, it's a dialogue. I've had, really, really interesting discussions with people with autism who are adults who do not like the idea of intervention, and it's tough. It's tough because many of the people that we encounter that are children, and we as a society, as a almost as a, I don't know if we've really thought this through, but we tend not to give children choice, whether the intervention is for social skills or whether the intervention is for learning algebra. We tell children what we want them to do and we expect them to do it. And I think that tends to be the truth. I think when you reach adulthood, treatment is, it becomes an individual's choice, what they think that they want to address. And again, I think when you think about autistic adults, it's variable. I mean, I will say we are not in the position to force anything on anybody, and we do see many adults in our clinic who are being evaluated for a diagnosis for the first time, and they're there because they have something that they want addressed. I mean, the people don't come, in my experience, people aren't coming to the clinic because of like an academic curiosity about whether they have autism. They're coming because they're experiencing some difficulties, and they're hoping that that understanding whether they have autism or not would be informative in addressing those difficulties. And so it's so it's really the issue that Sarah raises in her question is an important one, and it is it's hard to have rules. It's really hard to have rules. And so it has to be approached thoughtfully in the context of a dialogue, and it's relative and not absolute.
Great answer. Any other thoughts, questions? Jamie, this was a great talk, a great ending to our series this semester. Please stay tuned. Next year, we'll be starting a new series sometime in the fall, and we're also going to be having a series on autism and the law. And we have a number of other activities that we're going to be involved in doing in the fall, including potentially a drive-through with police for autism drivers who have licenses using the new blue envelope in the state of Connecticut. So stay tuned. Jamie, thank you very much. Any last words?
Two last words, not literally, two last statements. One is there's a question directly to me that Reyes missed the first part of the meeting. My understanding that this is being recorded, and that I think Eileen Farmer would be the best person to talk to. And then Sam just asked a question. Sam, I'm going to answer your question, but when you say you just got back from nap time, I mean, I don't know if I should be insulted that you slept through my talk or not. But
No, my son.
Oh.
I was watching your talk with my son, and he was getting very crabby.
No, I was thinking, how did you manage to sleep through my talk and yet ask such a relevant question?
I'm sorry, I put my son down for a nap.
The question is, have you seen the studies on the effects of transcranial magnetism with depression and anxiety? I have. And in fact, you happen to, I have manic bursts of research energy. And right now you've caught me in the midst of a manic burst about the potential of using TMS to treat depression and anxiety in people with autism. So Sam, I think, knows this from asking the question, but TMS is an FDA approved treatment for depression. I know less about anxiety, but for depression that has not responded to conventional behavioral therapy, cognitive behavioral therapy, or talk therapy, or medication. And so there actually are clinics at Yale, both in the medical school and at the VA, that are using TMS to intervene in treatment-resistant depression. And I'm very excited that it's not totally finalized, but I think it's very likely that a person's going to come to a postdoc with our group who's actually going to be splitting time between the treatment-resistant depression clinic at the VA and doing our autism work. It's a person with expertise in TMS, and what that person's goal will be is, and I hesitate to say as a therapy. I like to think of the idea of kind of studying it proof of concept as research before we would make the bold assertion that this is worthy of being called therapeutic. But hopefully this is something that we're going to start doing in the coming year, because I think it's a really, really exciting and relevant application that hasn't been looked at. One study, I actually checked, there's one study published that looked at treatment of resistant depression and autism with TMS, and it showed very promising results.
Got it. So just a side note for anyone who's interested in, I'm sure you've read this, Jamie, and you've probably talked with him himself, but John Elder Robinson actually wrote a book called Switched On about his experience with TMS. Yep. And how it affects him. So if anyone is curious about TMS and wants to read about a firsthand account, I highly suggest the book. He's a fantastic author.
I totally agree. And if you're if you've been too teched up to tolerate reading an actual book, there is also an episode of the Moth podcast in which John talks about his experience with TMS, which is also awesome. And he's like, it's worth it.
I know. It's just
anyways, and John's had a fascinating life. Just to give you a little teaser, he also has expertise in pyrotechnics and may or may not be the person who has designed flaming guitars for rock stars. But you'd hear about it in the podcast.
Specifically Kiss. He's the one who made the flaming electric guitar.
Yeah.
Okay. What a note to end on. Jamie, thank you so much. People will see you.
Thanks for having me.
Have a great summer. A fantastic talk. Thank you, Jamie.
Thanks everybody for your kind attention. Have a great weekend. Bye-bye.